So this table right here is showing us the temperature dependence of fatty acids based on growth temperatures of E. coli. And so this is original data coming from a paper from 1962 from the Journal of Bacteriology. And what this paper shows us is we're looking at the effective temperature on the composition of fatty acids in E. coli. This paper shows us a couple of different trials, or a couple of different experiments. Growth of E. coli at 10 degrees Celsius, 20 degrees Celsius, 30, and 40 degrees Celsius. Now, what we analyzed, or what was analyzed in this paper, was the presence and abundance of myristic acid, palmitic acid, palmitoleic acid, and oleic acid. So one of the things that this is kind of investigating is basically what do these E. coli, or what does this E. coli produce in certain growth conditions. Now, one thing that I want to draw your attention to is myristic acid, this short 14 carbon fatty acid. We had 4% for 10, 20, 30 degrees Celsius, and only at 40 degrees Celsius did you actually see an increase in the percentage of these short, fully saturated fatty acids. Now, what I want to do is ask you to think about how this information, how this data right here, which we'll get to in 1 second, how this correlates to what we've already seen. And one thing that I'm going to write out is the melting points of all of these different fatty acids. Okay, so we've already talked about these melting points, and I don't expect you to memorize these, but I do expect you to know the trends. Number of carbons goes up, melting point goes up. Number of double bonds goes up, your melting point goes down. Okay, so myristic acid has a melting point at approximately 52 degrees Celsius. Okay, palmitic acid has a melting point of 63.1 degrees Celsius. Palmitoleic acid has a melting point of 1 to 0.5 degrees Celsius, and oleic acid has a melting point of 13.2 degrees Celsius. And we've already kind of talked about this oleic acid and how, well, that really displays this information very well, because all things even, these are both monounsaturated fatty acids, but our number of carbons increased from 16 to 18, and we saw an increase on our melting point. Okay, so what I want to do is first look at our 10 degrees Celsius. Our 10 degrees Celsius shows us we have a high percentage of monounsaturated fatty acids. We've got a grand total of, what would that be, 64% of our fatty acids are monounsaturated fatty acids. 64% of the fatty acids here are either 16 or 18 carbons long. Then, if we jump all the way to our 40 degrees, well, 56% of our fatty acids are going to be, sorry, not polysaturated, they're going to be short, and they are going to be saturated. So then let's look back at, let's keep things the same. So looking at our monounsaturated fatty acids, we went from 64% of all the fatty acids produced by cells grown at 10 degrees Celsius to 21%. So we saw a huge reduction of these polyunsaturated, or no, monounsaturated fatty acids. I'm sorry about that. And so what this, I think, really speaks to is what's your pursuit, or what's the cell wanting? The cell is wanting a membrane to be fairly fluid. Okay, they don't want a completely rigid thing. And so at a low temperature, what do they have to do? They have to increase the percentage of monounsaturated fatty acids and reduce the number of saturated fatty acids. At a higher temperature, well, you kind of want to reach that same fluidity that we had earlier. And so as a result, we're not going to make as many unsaturated or monounsaturated fatty acids, because at 40 degrees Celsius compared to 10 degrees Celsius, you are considerably closer to these melting points. Okay, you're not at or above those melting points, but you're considerably closer. This is, you know, you're 10 degrees off of myristic acid's melting point, and you're like 23 degrees compared to where you were 40 degrees and 50 some odd degrees whenever you were growing at 10 degrees Celsius. So I want you to think about this as, you don't want the cell to be 100% fluid, or you don't want the membrane to be 100% fluid because you have to maintain some sort of structural integrity of that. But what this data shows us is that at a higher temperature, we have a greater preference for short and saturated fatty acids compared to at a lower temperature, well, you have a greater preference for monounsaturated fatty acids because of the fact that what they're going to do is they're going to give you, think about this as this approximately same level of membrane fluidity at 10 degrees and 40 degrees Celsius. All right, I hope this is helpful to look at that specific data. So lipid asymmetry is something that's very important to understand. Like, where are glycerose phospholipids only found on one side of the lipid bilayer? Are they found on both sides of the lipid bilayer? Because those are your mechanisms to communicate with things outside of the cell. Phospholipids are synthesized on or near the interior of the membrane. Now, there are two mechanisms, which we've already talked about, movement of these different lipids, but there are two mechanisms to get the phospholipids from one side of the membrane onto the other. So there's a class of enzymes, or there's enzymes known as flipases. Now, what they do is catalyze the movement or the flip-flop of specific phospholipids in what is known as facilitated diffusion. So what we've got up here is an uncatalyzed transverse diffusion. So a movement of lipid right here to the interior of the cell. The exterior of the membrane. So this is inside, this is outside. Okay, so this is uncatalyzed transverse diffusion. Now, that is very, very slow because basically if we're talking about a glycerophospholipid, we're talking about something that's arguably fairly polar, moving through a membrane that is extremely nonpolar. So to actually the energy to overcome that there's going to be a huge amount of energy required to overcome that resistance. Now, down here, we've got transverse diffusion, the same thing, but we're catalyzing that by an enzyme classified as a flipase. Okay, so that enzyme is going to equilibrate the distribution of the corresponding phospholipids. It's going to help overall with the composition and asymmetry of lipids on the inside and outside of those cells, or outside of those membranes. Phospholipid translocases are able to transport specific phospholipids across a bilayer in a process that is driven by the hydrolysis of ATP. So to overcome that energy barrier, you're going to use something like ATP and break a phosphodiester bond, that high energy phosphodiester bond, in order to drive that. Again, though, the movement of the lateral movement right here, so from this point to this point on our bottom example, that's just something that's going to happen very quickly and very easily without much energy input. Translocases can establish a non-equilibrium distribution of phospholipids in addition to a equilibrium distribution being kind of already present. Membrane structure and assembly, the phospholipid bilayer is known as a fluid matrix. The lipid bilayer is a two-dimensional solvent, and this just goes to show kind of an overall composition. One of the things that I think is most important anytime, and it seems very trivial, but I think that it's one of the most important things whenever you're talking about a lipid bilayer or a membrane in particular, you always want to orient yourself to the inside and the outside of the cell. Okay, so we've got our lipid bilayer right here. We've got outside of the cell, I'm writing in yellow then. Outside of the cell, inside of the cell. What are some of the things that we should sort of be drawn to? Now, what we have a huge abundance of is all of these glycerophospholipids and sphingolipids and everything else that we've already talked about, making up the vast majority of that. What else do we have? Well, on the exterior of a cell, we have what is known as a glycolipid. We have something known as a glycolipid. And a good example of that would be a ganglioside. These, well, first of all, GLYCO, glyco, you should be thinking about sugars. We've got a glycolipid where there are a bunch of sugars that are affixed to a lipid. So these hexagons here, think about those as your general structure for a sugar. Okay, so those are going to be on the exterior of the cell. So when you think about the movement of a glycolipid, chances are that's synthesized inside the cell, on the inside or the inner part of the membrane, but then you have an enzyme like a flipase that's going to move it to the exterior. Okay, then let's continue scanning across the outside. Well, here, this red chain right here, well, we got to follow that down, and this is an example of integral protein. This has a single transmembrane helix. So this is an example of a protein that is integrated into the lipid bilayer. So we've got one side of it on the outside of the cell and one side of it on the inside of the cell. Now let's go back outside the cell. We've got an oligosaccharide chain on a glycoprotein. So what this shows us is this is a protein, a membrane protein with an oligosaccharide. In fact, this actually has 4 different oligosaccharide chains. This is an example of a glycoprotein. Now, glycoproteins serve as great mechanisms to communicate with, or great mediums to communicate with other cells. But let's go back to looking at our polypeptide chain. This is an integral protein with multiple transmembrane helices. This has 1, 2, 3, 4 transmembrane helices. So this is a protein that spans the membrane. It is one polypeptide chain, but there are a total of 4 helices that span that membrane. Okay, now let's go on the interior of the cell, on the inside of the membrane. We've got a peripheral protein covalently linked to a lipid. Okay, so this would be a good example of post-translational modification that basically affixes a protein to a lipid. Okay. Next up, we've got a sterol on the inside of the membrane, but if you look at that sterol, it kind of looks like a hot dog. We've got those sterols kind of all over on the outside of the membrane as well. Okay. Then if we go, we've already talked about our integral single transmembrane helix protein. We've got another peripheral protein that's just kind of associated with the membrane. Then we got a phospholipid polar head group. Okay, so those are just our phospholipids. Those blue circles right here, those are our phospholipids, and those are the head groups that we've already kind of talked about. So when it comes to membrane composition, there is tons of stuff either in the membrane or simply associated with the membrane. We're going to dissect some of those components a little bit more, but some of these they should be fairly familiar because we've already talked about them. For instance, your sterols as well as a ganglioside, that would be an example of a glycolipid. Well, I hope this is helpful, and I'll see you in the next video.